Hi, good morning, good afternoon, good evening. So this is the second part of the model 3, which is study 3 and 4. In this, we'll be discussing about the diencephalon basal ganglia, the ventricles, and also the cerebrospinal fluid. So we are starting with the cerebellum, which is a small brain. So you can see it at the lower aspect, which is the red or orange niche is in color. Just as the name implies, it is called little brain, and it is very complex. Though it looks structural, morphologically it looks small. However, what it does is very similar to the cerebral cortex. It's also almost as large as cerebral cortex, but however, it is being folded and more thinner so that it will look smaller as compared to the cerebral cortex. And it contains about 50% of the neuron of the whole brain, so it's significantly similar. It's been divided into two hemisphere, and then we have one hemisphere sitting at the middle, which is referred to as the vermis. Okay, so in this image is not very properly seen. However, the vermis is what is located in the sagittal section, which is seen at the upper right corner. So the sagittal section, you can see it looks like two bumpy structure and a middle part, narrow part. So the middle narrow part also has a little part of a looking like a hemisphere. So you have two hemispheres joined by the vermis, and then it is also folded into several tiny folds that is referred to as the folia. And then after that, the deep sagittal section or the mid sagittal section, where we're talking about the superior image on the right hand side, the whitish coloration is what is referred to as the arbor or tree of life, because it looks like a tree. And that is mostly the whitish area is where we have the white matter, which is where we have the axons are blanketed or covered by a myelin sheet, mostly by the oligodendrocyte, and that is why they look whitish in nature. So this image also shows a section through the cerebral hemisphere, where the image on the left hand side is an actual section of a real brain, while the image on the right hand side is an illustration of what we have. So you can see what it looks. It's very similar to images, and then when you kind of zoom in, and then you can see that it looks like branches of tree. That is the arbor we are talking about. And in deeper area, you can see the white matter, which is referred to as the arbor. And then in the middle part, we have the two bumpy structures. In the middle line, we have what is referred to as the vermis. That is the hemisphere that connects the two cerebellar hemispheres. And deep also inside the white matter, just like I cycled in this illustration, we also have a grayish matter that is there located deep inside. So we have four of this collection of the gray matter. So the lateral one, the biggest one, is referred to as the dentate nuclei. It is very important because it serves as a major exit route for most of the neurons coming off from the cerebellar hemisphere, mostly going to the cerebral cortex, that is this big brain. Then deeper, you have what is called the globus, and then you have the vestigial, and you also have the embelliform. These are smaller nuclei that are found in the midline, and they are important, but for your class we're not going to discuss further about it. So here is an illustration, the one in green and purple, where the cerebellum is divided into three hemispheres actually. So you can see a purple color on top, the large green area at the back, and then a very tiny orange structure. So when you decide to spread it around, that is the other illustration on the right hand side of that structure. So you can see this is a flat illustration of the complete cerebellum, where you have the vermis in the midline, and then in the anterior purple coloration, that is where you have the anterior lobe, and then you have the posterior lobe is in green. So dividing the anterior lobe and the posterior lobe is what we refer to as the primary fissure. And then deep in underneath the posterior lobe, we have another orangey smaller looking lobe. So we have a midline looking area like the vermis, but this one is separated from the vermis. It's called the nodule. And then the one that looks like a butterfly wing on the lateral aspect of it, the same orange, it's called the flocculus. So it's like floating, like flopping its wing. So that's why it is called the flocculonodular lobe. So this flocculonodular lobe is important because it is associated with our vision. It allows us to, even when we are trying to turn our head, our body is balanced. Sometimes when you sharply turn your head, you feel like you want to fall down. So this folliculonodular lobe is important because it helps you in balancing as you are doing your visualization. And then this another image also shows the image on the superior aspect, where you see a bundle of green, a bundle of red, and a bundle of blue. Those are fibers coming into the cerebellum, coming in and out of the cerebellum, and that forms the connection with the anterior locating pons, because the cerebellum is located behind the pons and behind the medulla oblongata. So the pons is superior and the medulla oblongata is inferior. So you can see that some fibers are coming in from the inferior aspects, that is the red color. Some fibers are coming in with the green color and the middle, and then some fibers coming in the superior in blue color. So these are coming via different paths of connection, and they are called peduncles. So we have the superior peduncles, that is the blue ones, bringing in fibers in and out, mostly out, heading from the cerebellum, going out via the pons to enter the midbrain, and then they go to the top cerebral cortex. And then the green one mostly are fibers coming in from the pons. They are mostly pontine nuclei fibers, and that is why they are called the middle. They are coming in as the middle cerebellar peduncle. They are the largest peduncle. And then we have the last one, which is the inferior cerebellar peduncle, and this inferior cerebellar peduncle is taking in mostly information coming from the spinal cord or going towards the spinal cord. And then the last image on the bottom, which is having the yellow and green, is also just trying to explain the anterior lobe, which is separated from the posterior lobe by the primary fissure. And then we have a very small lobe, also represented in blue, which is the flocculonodular lobe, which is separated by posterior lateral fissures as well. So, and then on the anterior aspect, that is the pregnant looking pons. So this one is also the transverse section to show you the deep nuclei, which we already mentioned earlier on, but the color is indicating those deep nuclei. So you can see in green, that is the largest nuclei, which we said is the dentate nuclei, and the dentate nuclei, like I said, is the most important nuclei, which serves as a bypass for neurons leaving the cerebellum, mostly heading towards the cerebral cortex. And then the next one, we have the interposal nucleus. So the interposal nucleus are referred to as the embryoniform and globus nuclei, and then the most in the midline is referred to as the vestigial nuclei. So for your class, we are not going to discuss much on those deep nuclei. Then, like we said, mentioned earlier, we have the middle and superior and inferior cerebellar peduncles, and like I said, mentioned earlier on, the superior peduncle is connecting the cerebellum to the midbrain, and it takes up the information up the way to the cerebral cortex, while the middle cerebellar peduncle connects the pons to the cerebellum, and primary carries input information from the pons to the cerebellum, just like we mentioned earlier on. So these are the colors representing that. So the green is the middle cerebellar peduncle, the red is indicating the superior cerebellar peduncle, while the blue is the inferior cerebellar peduncle, which connects the cerebellum to medulla oblongata. Like I said, carries in and out information to the cerebellum. All right, so the functions of the cerebellum is to control our voluntary movement. So it does the balancing. So cerebellum is important for balancing any motor movement. So when we are walking, if our cerebellum is affected, we cannot walk balance. We just feel like we are dizzy, we want to fall down. So it does that. And then when we are stepping down, because we know the distance, like when we're going down the steps, we know the distance which we are going to, and then we know how to contract the muscle, so that are we contracting it too much? That is, are we lifting the leg too high, or we are just lifting the leg too slow? And are we relaxing the muscle at a very high and intense tone, or we are relaxedly going down? So that is why some people say, oh, they missed themselves step, because maybe they didn't calculate the distance between the step, and also that affected their gating, that is their normal movement. So it affects your brain from calculating how hard you need to contract the muscle, or how soft you need to contract the muscle. Another thing is that it's important for cognitive function. So this cognitive function includes more paying attention, more our walking memories. Like, okay, we know sometimes when you are walking, you are used to steps. You don't even need to look at it. You just continue walking without necessarily looking at where you are going to. This is because of the collaboration between the cerebellum and our memory centers, which is referred to as the hippocampus. And next, we are going to talk about the injuries or lesions to the cerebellum. If one has a lesion to the cerebellum, mostly the anterior, so the anterior lobe is involved in the control of our trunk, that is our chest and abdominal region, as well as our back. We are going to have challenges with our posture, maybe not straight, maybe we feel curved, and that is where you are not balancing your right and your left side, and which is referred to as ataxia. And then next, the posterior lobe is more involved with control of your upper limb. And upper limb, because of our skill, the reason why it's bigger than the anterior lobe is because of how our fingers are skilled in writing. So we need to balance our upper limb as much when we are holding something, holding a bucket, trying to lift a weight, etc. So it is important to be able to maintain that structure. While flocculonodular, like I said earlier on, is highly involved with our eyes and the locomotion of our eyes, that is movement of our eyes. I know sometimes, most of the time, when our eyes are moving, it tends to also move with our head, and that is why the flocculonodular lobe is associated with the movement of the eyes and the face as well. So if we tend to have injuries to any part of the cerebellum, we have different association or condition associated with it. Like I said, it is also assisting in our smooth or motor movement. So if we have a lesion, definitely we know the movement, our motor control is not going to be smooth, and that is why we have the symptoms of a tremor, and also we have slurring in our speech because of the flocculonodular, which also assists in control of the muscles around our vocal cords. So now we're going to talk about the diencephalon and basal ganglia. So the diencephalon is a collection of deep nuclei found deep within the cerebral cortex or cerebral hemisphere. Please, cerebral hemisphere is different from cerebellar hemisphere. So cerebral is small as a big brain, cerebellar is a small brain. So now we're talking about the big brain. And so you can see the diencephalon is that deep purple looking area inside the cortex, and you can see below it is the green area. So the green area is the brainstem, which houses the midbrain, the pons, and the medulla. So above the brainstem, we have what is referred to as the diencephalon. So these diencephalon are also referred to as the thalamic family, where we have the thalamus, which looks like egg in structure. So in the image below, you see the coloration. So you see the thalamus is mostly chunky, and we have two of it, one on the left hand side and one on the right hand side, and the two of them are connected by what is referred to as the interthalamic adhesion. Next, anterior and inferior to the thalamus, so on the front and below the thalamus, you have what is referred to as the hypothalamus. The hypothalamus is a complex collection of nuclei that controls several hemostatic activities of our body, including our endocrine system. So we're going to talk about it later on when we talk about the endocrine system. It also helps in controlling a lot of our autonomic nervous system, and also collaborate with different parts of the brain in coordination in maintaining balance. Another thing it does is also helps in our vegetative centers, that is our cardiac centers, respiratory centers, and also it helps in controlling our taste, how we feel dehydrated and how we feel taste, how we feel hungry and how we feel satisfied. So a lot of those are being controlled by the hypothalamus. Next, we have what is referred to as the epithalamus. The epithalamus in the image below, you can see, is denoted by blue. So the epithalamus are a collection of two bodies. We have the two nuclei found on one right and left. So at the middle of it, you have the pineal gland, which is also very important. So the pineal gland is very important for controlling our sleep and awake cycle. So if we're sleepy, if it is dark and it's cold, we want to sleep. But when the light is there and it's very hot, we do not want to sleep. We want to remain awake. So that is the main function of the pineal gland, by releasing what is referred to as the melatonin. Next, we have the subthalamic nuclei. Just like the name implies, it is found, these are gray matter found below the thalamus, and that is why they are called subthalamic nuclei. So in this image, you cannot appreciate it much. It's when you have a section through the midbrain, as we're going to see later on, that you're going to see a little bit of the subthalamic nuclei. And lastly, we have the metathalamus. So the metathalamus are two bumpy structures found on the lateral aspect of the thalamus, and they are called the lateral and medial geniculate body. So we're going to see them later on in the images as we move on. So the image on top there is, you can see something like a snake coiling. The top on the left hand side is an image where the upper part of the brain has been removed so that you can appreciate the deeper structures. So the two egg looking structures on that on the midline, okay, is what is referred to as the thalamus. So that's right and left thalamus. In between the two of them, we have a space, a narrow space, very small space, that is called the fourth ventricle. And then and also just behind it, you can see two bumpy structure with another small projection in that aspect, that is referred to as the one on the superior aspect, looking very smallish. It's called the pineal gland, while the two bumpy structures on the lower aspect are called the superior colliculus, which are structures found on the posterior surface of the midbrain, like we spoke about earlier on. So the top right image is a coronary section into the brain, and then you can see the two thalamus right and left, where you see the purple color in the midline. So you can see two thalamus right and left, and then a very small or narrow space in between them. So the two thalamus are connected by what is referred to the interthalamic adhesion, and the space between them is referred to as the third ventricle. And then you can see below it, you have a very light green color, which is referred to as the subthalamic nuclei. That was the nuclei we couldn't see earlier on. Like I said, it is found at the bottom of the thalamus. Then on the image on the bottom aspect is where you can actually see a white curve looking or sickle looking structure, and then on the bottom of it you can see like something reddish like a line, and then you can see something oval below that. That is the thalamus, and then you can see the white round structure there in the midline. In the middle is referred to as the interthalamic adhesion, where the two thalamus connect. And the ventral aspect, you can see the optic chiasma, and then you also get to see the hypothalamus located behind the optic chiasma. Similarly, as you go down below that white curved structure on the right hand side, you can see a small projection. That small projection is referred to as the pineal gland, and like the one we said produces the melatonin to assist in our sleep and wake cycle. And then next below that, you're going to see the superior and inferior colliculi, structures that are found on the posterior surface of the midbrain. Here also in the first image, the small image, we have the coronary section, and the green highlighted area is the thalamus, and in between there are two thalamus, and in between the two thalamus we have what is the interthalamic adhesion, and then in between them is a very narrow space that is referred to as the third ventricle. Superior to the thalamus, you have like a band or going upwards, that is the corpus callosum, and just before the thalamus, between the thalamus and the corpus callosum, is a very almost a U-looking shape space. Those are part of the space of the lateral ventricle. All right, so in the image, the enlarged image, the blue at the top part, where the blue coloration is where we see the diencephalon, and then you can see it is well labeled, where you see the epithalamus, including the posterior commissure, the habernuclei, as well as the pineal gland. And then ventral to it a little bit is the thalamus with the interthalamic adhesion, and most ventrally you can see the hypothalamus, and inferior to the hypothalamus you see parts of the pituitary gland. And then in the lower image is an enlarged image of the thalamus, where the thalamus is subdivided into various nuclei collection. And so you don't necessarily need to remember this for your program. However, I just wanted to show you that on the right hand side of the two thalamus that have been shown with the arrow, there is a green area that wrote G and LG. So that is the lateral geniculate and the medial geniculate bodies, which are all part of the metathalamus. So here is also just an included part where we talk about the major part of the nuclei. But like I said, these are a collection of nuclei or collection of cell bodies performing similar function found inside the thalamus. So the thalamus is a structure, is largely a structure, but has different sub collection of nuclei, which you don't necessarily have to remember. This I just added for those people that want to read, but this you don't necessarily have to remember the individual nuclei. So the functions of the thalamus, like I said, the thalamus is very important because all sensory information has to come and change the button. So it's just a relay station where buttons are exchanged between sensory information. So like I said, all sensory information come to the thalamus before they move, before they synapse with another neuron, which goes to the different parts of the hemisphere to tell them the information they've gotten from the exterior. So sensory information are afferent, AFF, information that comes from the exterior, and they go through the brain via the thalamus. So the only sensory fibers that is not coming directly to the thalamus is the olfactory sensory system. So the olfactory sensory system is a system that uses to perceive smell, and so for it comes in from the olfactory nerve, and then comes in as olfactory tract, and then from the synapse inside what is referred to as the amygdala. So in this image, we don't have the amygdala, but amygdala is important because it is associated with our memory, and that is why whenever we smell something, we remember something associated with it. Like when we remember the first time we had jollof, or there's a specific way our mother cooks jollof, and when we perceive the smell, we'll be like, oh, we remember our mother old good times. All right. So next is the hypothalamus. Like I said, the hypothalamus is a small but vital region of the brain that is important for our survival and several functions. So some of the nuclei in the thalamus include the mammillary body. The mammillary body is very important because it's associated with the amygdala and also associated with the hippocampus. And like we said earlier on, hippocampus is a part of the brain that is important for memory formation, both long-term and short-term memory. So the mammillary body connects directly with the hippocampus via the fornix. And there we have the infundibulum, which is the connecting part of the projecting part of the pituitary gland. We have the optic chiasma and other nuclei. So you don't necessarily need to remember these individual nuclei. Personally, I would do not ask you about certain a nuclei, except is part of the pathway that we are going to discuss in a further class. So next, we're going to talk about the functions of the hypothalamus. Like we said, hypothalamus is important in temperature regulation, important in hunger and thirst. Also, it correlates or interacts with the pineal gland to help us in our circadian cycle or sleep-awake cycle. Like I said, we're going to see a lot of it in the endocrine system when we talk about the endocrine system, which is module 2 of this program. Then it also controls the autonomic nervous system, and because it's also very close to the amygdala, it also helps in controlling or interacting or expressing or relating our emotions. So next we're going to start another system, and that system is called the basal ganglia. So the basal ganglia is important ganglia where it's formed by a connection of various nuclei. And apologies, I didn't talk deeply about pineal gland. It's because we're going to see pineal gland later on when we talk about the endocrine system. We didn't talk further much on hypothalamus because we're going to talk about hypothalamus also in the endocrine system. And then we are going to also talk about the thalamus in this when we talk about the basal ganglia. This is another collection of nuclei, and also we're going to see the subthalamic nuclei, which we are also seeing in this particular collection or complex connection of nuclei in the encephalon. So the basal ganglia is a connection of some deep nuclei that helps in controlling or modulating our voluntary movement, our voluntary motor movement. So it helps in initiating, coordinating, controlling our voluntary movement. So that is what the basal ganglia does. And the basal ganglia is a collection of nuclei found deep to the cerebral hemisphere. So you can see them in different colors here, and they connect in two pathways. They connect to perform two functions. So one is to initiate and increase the movement, and the other one is to actually dampen, reduce, inhibit, or stop a movement. So direct pathway initiate and enhance movement, while indirect pathway, what it does is dampen, inhibit, or stop movement. So like I said, that the nuclei, collection of nuclei that forms these basal ganglia are several in number. So from this image, you can see this one is the lateral view. The first one on the left hand side, the lateral view, and the one on the right hand side is posterior lateral view. So it means you're looking at both the posterior and also the lateral view. And so for the lateral view, only the lateral view, so you can see the purple snake looking structure and the green horn structure that goes round and then goes to attach to something at the bottom. So the large blue structure, or the head of the snake, looking like a head of the snake, is what is referred to as the putamen. And then the green structure that goes round around like a horn is called the quadrate nucleus. And then at the end of this quadrate nucleus, at the end of the tail of the quadrate nucleus, is small blue structure, that is the amygdala we're talking about earlier on, that is involved with memory and emotions memory. So deep to that purple on the blue structure, the putamen, we have another purple structure. So there are two layers of this purple structure, but from this image you can maybe just see one, not very clearly. So that is referred to as the globus pallidus body, which we have two layers. You have the globus pallidus externa and globus pallidus interna. And so the two of them are also perform two similar functions, but on two or different organs. And then deep inside that, you can see a pinkish looking structure, that is the thalamus. So basically from this image, some of the structures that interact to give rise to the circuit or the basal ganglia. So on the posterior lateral view, you could also see more structures contributing to the basal ganglia. So you can see deep down at the bottom on that is the image on the right hand side, which is looking at the posterior lateral view. So from the bottom of the small brain, you can see those deep nuclei we talked about earlier on, the dentate nuclei. So the dentate nuclei also contributes. Like we said, it helps in controlling our motor movement. It helps in balancing our gait so that one side we don't put a heavy step as compared to the opposite side. We are moving at a balanced ratio. One side is not pulling the other because the two sides of our body is trying to balance, and that balancing is by the cerebellum, and these fibers are coming via the dentate nuclei, and so that is why we see the dentate nuclei. Then at the superior aspect, we see another blue structure. This blue structure is what is referred to as the substantia nigra. So substantia nigra, just like the name, is a dark substance, and that is why you have the substantia nigra. So this dark substance found in the midbrain is very important because it produce what is referred to as the dopamine, or the dopagenic neuron. It has the dopagenic neurons, and they produce the dopamine. So these dopagenic neurons are blackish in color. That is why their name is substantia nigra, and they produce dopamine. So these dopamine are important because they go and bind to certain neurons to enhance the expression of some things. All right, so that is the nuclei from the posterior aspect. When you go above it, you see the purple structure that goes around it. That's the quadrate nucleus. And then the green in this image, on the left hand, on the right hand side, the green here is the putamen. All right. So these are the components of what we discussed. So the striatum is a combination of the quadrate nucleus and the putamen, while the globus pallidus, we said we have the globus pallidus interna and globus pallidus externa. And then we have what is referred to as subthalamic nuclei. You remember that is the nuclei, the thalamic family, or the diencephalon family, that is found at the bottom of the thalamus. And then next we have the substantia nigra, which is the darkish coloration neurons, dopagenic neurons found in the midbrain. And then then you also have the ventral anterior and ventrolateral nuclei of the thalamus. And then we also have the dentate nucleus of the cerebellum, which I didn't put here. So based on that, we can see that this organization of the structures is that the corpus luteum and the lentiform nucleus. So the lentiform nucleus is a combination of the putamen and the globus pallidus. So the two globus pallidus, because they lie over each other and they give rise to that form shape like an L, or a curved shape like an L-shaped structure, and that is why they are called the lentiform. So you have the putamen on the exterior, and then you have next when you have the globus pallidus externa, and then deeper you have the globus pallidus interna. Then curving above them like a horn is what we have the quadrate nucleus. So however, the quadrate nucleus and the putamen are called the striatum, and then the putamen and the globus pallidus interna and externa together, they are called the lentiform nucleus. So that is why you can see that. And other structures that form the basal ganglia, you also have the subthalamic nucleus, substantia nigra as well. So here on the image above is the different parts of the lentiform and the basal ganglia structures, which are well labeled. And then the image below that large one on the left hand side, you can see this is a cross section through the midbrain, where you can see the substantia nigra in the anterior aspect, where you see the dark collections of neurons, and also it's been labeled the pars compacta and pars reticularis. So the pars compacta is the deeper part, or which have the thicker nigra for neurons, while the pars reticularis fibers are the parts of it, the anterior part, where it's very light, having less neurons. And then on the right hand side of that bottom image, we have the different nuclei of the thalamus. But like I said, for these we are only interested in the ventrolateral and ventroanterior nuclei of the thalamus. So just to remember or give ourselves an orientation, the first image is a coronary section, the second image is a mid-sagittal section, and then the third image is a transverse section, that is looking from above. If you cut it from above, that is what you see. Coronary section is just like cutting your body from right to left, and then this sagittal section is cutting it to via your nose, midline of your nose, and that is what it means. So here also similar to what we've presented earlier on, the image on the most top area is just an indication how the lentiform nuclei, which is the putamen, globus pallidus interna, globus pallidus externa, is separated from the quadrate nucleus by what is called the internal capsule. So the internal capsule are white matter fibers extending from the cerebral hemispheres going down to the spinal cord, and that is why they are called the corticospinal fibers, but at this point they are referred to as the internal capsule. So still giving us different orientation of the structures we just saw. So the image on the right hand side, the one on the bottom, is just trying to give you a coronary section through the brain, and then you can see on the lateral aspect you have the putamen and globus pallidus, which forms the lentiform. On the top part is also on orange, is called the quadrate nucleus, which is located very on top the thalamus, and then you can see the thalamus in blue, and below the thalamus you have the red nucleus, and then you see the subthalamic nuclei, and then in front or below the subthalamic nuclei you see a darkish dark or black coloration, that is the substantia nigra. So this is also anatomical orientation, how you'll be able to identify the structure. This is the coronary section of the brain. This is a transverse section or horizontal section, where you look at it from the superior aspect, where you can see part of the quadrate nucleus, and then you can see the lentiform nucleus, that is the putamen, and then the globus pallidus interna and externa. Then here you find the on the middle aspect you have the thalamus. Alright, so this image is showing you in green color how the internal capsule or the corticospinal fibers are going down from the cerebral hemisphere all the way to the thalamus. So you can see them going downwards. You can see them as the cerebral peduncle. So the cerebral peduncle is different from cerebellar peduncle. So earlier on we talked about cerebellar peduncles. Those are hands for the small brain, while this one is a hand for this big brain. All right, so you can see the fibers that are stained green are coming from the cerebral cortex, and they go into the midbrain as the cerebral peduncles, and then pierces the pons, and you can see them inside the medulla oblongata, and they form what is referred to as the pyramids. Those give rise to the two bumpy structures and that area. So here is what is called the internal capsule. So here is also the sections, and for you to understand the location of the structures. And this is what we were talking about: the internal capsule extending from the cerebral hemispheres, goes into the midbrain as the crus cerebelli, enters the pons, and then they enter into the medulla oblongata as the pyramids, and majority of those fibers, like 85% of them, crosses to the opposite side of the brain. That means the right side of the brain controls more of the muscle or motor control on your right hand side. Well, yeah, so like I say, the fibers coming from your right hand side do more controls to the body structures or motor control on your left hand side. That is why 85% of the fibers have crossed and they do that function. So here is just what the basal ganglia does and the movement. So the basal ganglia, like I said, they are a group of subcortical. So when we say subcortical, we mean below the cerebral hemisphere. That is why they are called subcortical, and they are involved in inhibition and controlling voluntary movement, as well as other functions like learning and reward. That is why it helps you in like learning of dancing, learning of writing, learning of singing because of using your vocal cords, learning to do some of your motor motions as well. And also reward: the moment you are able to do dance, you automatically clap your hands without you knowing. So those are kind of reward centers. They operate through two pathways, like we said earlier on, where we have the direct pathway, which helps in facilitating movement, so initiating and facilitating movement, and we have the indirect pathway. This pathway generally inhibits movement, suppressing unwanted movement. So the functions, like we said, of the basal ganglia is to have a motor control, to initiate and control voluntary movement, and also assist in motor learning because of its proximity to that structure, which we said the amygdala and and hippocampus. And then it's also the cognitive function is to execute functions such as planning, making decision and problem solving, reward and motivation, and also emotional processing. So the direct pathway is a pathway that comprises of the cortex, the putamen, and also we also have the quadrant. So the quadrant and the putamen, we said, is referred to as the striatum. So you can see it on with purple. And then we also have the external pallidus externa and globus pallidus interna. Then we also have the substantia nigra as well as the thalamus. So, and then you can see those are structures that mix up the basal ganglia. So for the thalamus for the direct pathway, at a default, normally thalamus is excitatory. Thalamus is always sending information to the cortex to execute emotion, to execute emotion, to execute emotion. So if on normal basis our thalamus is allowed to be doing that, our body will always be vibrating, will be shaking, even if we don't intend to move. However, we have what is referred to as the globus pallidus interna. So the globus pallidus interna has a direct hold onto the thalamus. It means inhibit naturally the thalamus. So on a default, like I said, if we're all sitting down, our body is not moving, it means that the globus pallidus interna is holding on or inhibiting the thalamus from moving. Okay, so it is sending inhibitory neurons to the thalamus, and the thalamus cannot send excitatory neurons or excitatory information to the cortex. All right, so that is the second step. Then if you want to execute emotion, if you want to move, what happens is that excitatory neurons, referred to as the glutamate neurons, come from the cortex. So after we've made the decision that we want to move or we don't want to move, either way, the cortex sends the information to the striatum. So the striatum, like we said, is the caudate nucleus and the putamen. So these two tends, when the cortex sends an excitatory information to the striatum, then the striatum is going to send inhibition. So striatum is going to prevent globus pallidus interna from acting on thalamus. So if these fibers that is sending from caudate nucleus and putamen, which are the striatum, to the globus pallidus interna, are inhibitory neuron, or we call them GABAergic neurons. So when the caudate nucleus and putamen inhibits the globus pallidus interna, it means that globus pallidus interna cannot prevent thalamus from exciting the cortex. So gradually it release or loosen the hold of globus pallidus interna on top of the thalamus, and thereby the thalamus can actually be activated and then goes to excite the cortex so that emotion could be executed. But because you want to maybe increase the tension or contraction of your muscles, maybe you want to move your hand very forward. So now you've initiated to raise your hand, but you want to extend, you want to move the hand forward. It means that you need more power. That is when you have what's referred to as the substantia nigra comes into play. So substantia nigra tends to stimulate and then release, is being stimulated by the subthalamic nuclei. So the subthalamic nuclei recognize that the globus pallidus, we need more movement. So the subthalamic nuclei stimulates the pars compacta, pars reticulata of the substantia nigra, which is that dark area, and that fibers tends to send in neurons or excitatory neurons that goes to increase. So it goes to excite more that GABAergic neuron that is being released from the caudate nucleus to caudate nucleus and putamen that goes into the globus pallidus. So it means it increases the activity of the GABAergic neuron that is going into the globus pallidus interna, increasing the hold on the globus pallidus interna, and thereby releasing more of the thalamus, and thereby the thalamus will continue or increase its activation of the cortex, where the cortex is being told, okay, execute this motion. So that's is how the direct pathway occurs. So if you want to also increase the movement, if you're running, you want to increase your pace, the subthalamic nucleus goes to stimulate the substantia nigra, that is the pars reticular of the substantia nigra. Then these fibers goes to bind on top of the excitatory neurons coming from the caudate nucleus and the putamen. So this inhibitory excitatory fibers goes to bind on that GABAergic neuron on a receptor known as the D1 receptor. So this D1 receptor is important because it tends to excite. Later on, we are going to see the D2 receptors, which is going to do slightly opposite to that. So the D1 receptors bind to the GABAergic neuron, leaving the caudate and putamen that goes to the globus pallidus interna to prevent the globus pallidus interna from inhibiting the thalamus. Thereby the thalamus now is more free and more excited and sending more excitatory activation to the cortex and telling the cortex, I need you to move to execute the motion more. So this is direct pathway and how to enhance the direct pathway. So the first cycle was when the caudate nucleus inhibited the globus pallidus and slightly the thalamus activated, that was initiating the movement. However, increasing or modulating increasing the movement or facilitating the movement was that when the subthalamic nuclei excited the substantia nigra, which substantia nigra sends a neuron that is an excitatory neuron that binds to the GABAergic neuron leaving the quadrate nucleus and putamen and binding via the D1 receptors, increasing their activity, which means it increases its inhibition on the globus pallidus interna. I hope you understand that. So next we're going to be talking about still talking about the direct pathway. This is to show you how the substantia nigra pars compacta, sorry, it was the pars compacta, not the pars reticularis, but the pars compacta, you can see in this image, it goes to bind via what is called the D1 receptors, and D1 receptors tends to increase its inhibition on the globus pallidus interna, and the globus pallidus interna reduces, so that X at the bottom there indicates that the globus pallidus interna can no longer inhibit thalamus, and thereby thalamus can continue to activate or increase its own pathway. So now we're going to be talking about indirect pathway. So the indirect pathway, we said, also starts from the striatum. However, its pathway it's a little bit slightly out of the road. It doesn't go to the to excite the globus pallidus interna, but rather it goes and inhibit the globus pallidus externa, and the globus pallidus externa, which was holding on to subthalamic nuclei, that is STN in this diagram, is then inhibited. So let's start from the beginning so that we understand this part of the pathway. So like we said, that the substantia nigra, the subthalamic, the globus pallidus interna normally has a hold or inhibits the thalamus alongside the substantia nigra reticularis, which that is the SNR. So normally the substantia nigra reticularis is also supposed to inhibit the thalamus, but in this pathway, what happened is that the information the intention to reduce the movement is being sent in by the cortex to the striatum, and the striatum, what it does is now sends inhibitory fibers to the globus pallidus externa, which naturally was supposed to inhibit the subthalamic nuclei. So by the time you are able to stop, like when you send the excitatory to the striatum, don't forget, is globus pallidus, is putamen and the quadrate nucleus. So these two send fibers to the globus pallidus externa, inhibiting the globus pallidus externa, because naturally the globus pallidus externa inhibits the subthalamic nuclei, which naturally subthalamic nuclei is supposed to activate, okay, is supposed to activate or excite the substantia nigra, and because now you cannot now the effects of the globus pallidus interna, that externa, GPE, is being inhibited. It means that subthalamic nuclei, which is STN, is now excitatory, which tends to stimulate the substantia nigra reticularis, which is SNR, and then substantia nigra reticularis, which also sends it's supposed to inhibit the thalamus, inhibits the thalamus from activating the cortex. To increase this inhibition, subthalamic substantia nigra pars compacta also sends another fiber referred to as the D2 receptors, and these D2 receptors tends to increase the inhibition on the globus pallidus externa, and the globus pallidus externa now cannot hold or inhibit subthalamic nuclei, and their subthalamic nuclei will also keep exciting substantia nigra reticularis, which tends to increase its hold on the thalamus. So this is how the inhibition is being done. So it reduces unwanted movement. That is why when we are moving our hands go straight without vibrating, and also when we want to stop movement, we can actually hold it gradually or hold it abruptly, depending on how our brain decides. So like we said, The D2 receptors coming from the substantia nigra pars compacta expresses D2 receptors, and these neurons are part of the indirect pathway, and which binds to increase it's the inhibitory action of the striatum on the globus pallidus externa, and thereby these cannot prevent the subthalamic nuclei from inducing or stimulating the substantia nigra reticularis, which tends to inhibit the thalamus. So this is how the indirect pathway inhibits the movement. So now we're going to talk about the venous drainage. So the venous drainage, mostly they drain into the straight sinus, and also after from the straight sinus they drain into the transverse sinus, and from the transverse sinus they drain into the internal jugular vein, which then goes to drain into the right atrium via the superior vena cava. So next is the arterial supply. The arterial supply are coming from the middle cerebral artery. So the middle cerebral artery is a continuation of the internal carotid artery. So the moment it reflects, is divided into two. One becomes the middle cerebral artery. The other one continues arterially as the ophthalmic artery to supply the eye structures. So arterial supply, I said, is very significant because when you have a blockage, it tends to result to different types of stroke, or when you have a rupture of these arteries, they will also end up with hemorrhagic stroke. So like we said, clinical significance of basal ganglia: if there is a lesion or injury to any part of these nuclei which we talked about, that is the thalamus, the quadrate nucleus, the putamen nucleus, the lentiform, which is the lentiform is the putamen on the exterior, globus pallidus externa and globus pallidus interna. Or most of the time when you have degeneration of that dark neuron, which we said the substantia nigra, when you have those kind of lesions and injuries or reduction in their sizes, they lead to different type of neurological diseases. One of the most common is the Parkinson's disease or Parkinsonism, where it is characterized by tumors, rigidity, slow movement, and difficulty initiating a movement, which we've all seen here. Another one is also a highly genetic condition, where it can start as early as at 16, 17, even younger than that, as compared to Parkinsonism. Parkinsonism is associated with neurodegeneration associated with age. When we are getting older, it could be genetically, but manifestation is more older than in younger persons. However, the Huntington disease is also characterized by also involuntary movements, like we can also have reflex and jerking and everything, cognitive decline, and emotional disturbance, because you remember the location of the structures to amygdala as well. So the Huntington is, like I said, is more of a hereditary kind of condition, and this the manifestation is very early in life. Even as people younger than teenagers can get it. And then you have the dystonia, which is also characterized by sustained muscular contraction. So you're supposed to, your body is very stiff, and then you find out that you're flexed to a particular position out of the normal midline and balance structure. So the symptoms of the Parkinsonism, like we said, mostly commonly is the tremor, slowness, rigidity, difficulty with balance and coordination, marked facial expressions. You cannot identify. There's no emotion in the facial expression. There is also soft speech and also stumped posture. Most of the time, like we said, it is associated with the degeneration of the substantia nigra, which are the dopamine producing neurons. So you can see in this image where the substantia nigra, the one on the left hand side is a healthy midbrain, while on the right hand side you can see a significant loss of the dark neurons or the substantia nigra neurons. While in the Huntington disease, like I said, we have the chorea, which is an involuntary jerk movement, like we described earlier on, impaired coordination and balance, difficulty in speech and swallowing, cognitive impairment, including the early dementia in very young individuals, and depression, behavioral changes as well, And this is because of a CAG nucleotide repeat. More than 35 repeats are found in the genetic makeup of an individual. And then when we talked about the dystonia, it is where the muscles are hyper contracted, and you can see in this particular individual where the voluntary muscles are hyper flexed, and thereby leading to this distorted body proportion. So another condition also associated with the basal ganglia is the multiple system atrophy, which is called the MSA, and it's also highly associated with genetic factor, but we also have various environmental factors including plastic waste, pesticides. So Nigerians have started seeing a huge increase in neurodegenerative diseases, and people are believed to associate it with like the flaring of gas, and also associated with fertilizers in the farm. However, we still haven't identified what is our prognosis or what are the risk factors here in Nigeria. I believe some of you can use that as part of your research, your final year research. So there are more type of conditions associated with the basal ganglia, and you can read the sum of it. So for treatment, most of this we do not have a complete treatment, but rather we have something to maintain these particular conditions, like the for dopamine agents. Dopamine is a very large size of a protein, and it cannot pass the blood-brain barrier of the brain, and thereby you could see that they decided to design another agent which is very similar to which acts similar to dopamine, but it is very different from dopamine. It is called the levodopa, which basically is an agonist which helps in increasing the function of dopamine in the system. Then for the Huntington is also medication to manage the chorea, that is the jacking symptoms and other symptoms. And dystonia, you also have injections and medications to assist in the relaxing of those particular muscles. Others are the deep brain stimulations, where sensors are put into the different parts of the brain to stimulate and to regulate the activities of the brain. Others include physical therapy to strengthen muscles and also enhance the memory of those muscles, and also occupational therapy, which helps individuals maintain independence in their daily lives, and also speech and language therapy to improve their talking. So next, we are going to talk about the ventricles and cerebrospinal fluid. So the ventricles are spaces found inside the brain, and also you can see some of them extending into the spinal cord, which are referred to as the central canal. So these ventricles contain a fluid referred to as cerebrospinal fluid, and this cerebrospinal fluid are very important because if you notice, the tissues of the brain do not have blood vessels directly going into them. So one of the ways they can have transportation or easy diffusion or transportation of nutrients and food and other necessary agents to the brain, and also waste material out of the brain, is via the cerebrospinal fluid, which our brain is swimming into it. So internally we have the cerebrospinal fluid inside the brain, which helps to reduce the weight of the brain, which is referred to as buoyancy. However, outside of the brain also we have the brain is also covered by a layer which is called subarachnoid space, and the subarachnoid space also allows for the cerebrospinal fluid is located there. So cerebrospinal fluid are produced inside the ventricle. They flow inside the different parts of the brain, that is interior, and also the spinal cord, and they leave these parts of the brain to go into the subarachnoid phase via a foramen referred to as lucica, and also we have another foramen magendi, which we're going to talk about them later on. So this is a coronary section of the brain, and then you can see the lateral ventricle ventricles. And so the lateral ventricles are the giant ones. So the first image on A is a ventral view of it, where you see it looking like a horn structure, and the one, so two horns at the lateral ventricle. The one at the midline is called the third ventricle, which goes down via the aqueduct of Sylvius, that is a narrow passage, and enters the fourth ventricle, which is that diamond shape in structure, those blue areas, or the underneath blue areas. The next image is a lateral view of it, and then you can see how the horn extends all the way to the occipital part of the cerebral cortex. And also you can get to see that in the middle line, where we have the third ventricle, you get to see a white structure in the midline. This is where the two thalamus tends to fuse together, via known as the interthalamic adhesion. And then down it goes narrow, and then goes into the fourth ventricle. Then after the fourth ventricles, it goes down very narrow into the central canal that extends into the spinal cord. And then down there as well on the lateral view, you can see what is referred to as the lateral aperture. So the lateral, we have two lateral aperture and one median aperture. So the two lateral apertures, they are called the foramen lucidus, which allow for the cerebrospinal fluid to leave there to enter to the subarachnoid space, which we are going to see later on. And then we have one in the midline. It is called, so you can see it up there, we have the median aperture. So the median aperture, we only have one on the posterior aspect, and it is called the foramen magendi. So like we said, the ventricles are interconnected cavities within the brain containing the cerebrospinal fluid, and you can see the ventral view and also the lateral view of these structures. And then you get to also see the coronary section, where you get to see the two lateral ventricles at the midline, a narrow area there, which is the third ventricle, because this is a ventral view. That is why you look at it very small. And then you see the narrowing between that third ventricle, between the third ventricle and the fourth ventricle is a narrow, very narrow canal, which runs inside the midbrain as well as the upper part of the pons, and empties into the ventricle, which is located posterior to the pons on the above and medulla oblongata, however located anterior to the developing cerebellum, which is a small brain. So like we said, we have the lateral ventricles, which are the largest one, and you can see them, they have a horn shape, they have we have the frontal, temporal, and occipital horns. That is why you see that on the lateral view, you see another horn extended to the back. On the third ventricles is located in the midline between, so below the corpus callosum and then between the two thalamus. So they connect also to the lateral ventricle via what is called the interventricular foramen. So that is, the lateral ventricle connects to the third ventricle via what is referred to as interventricular foramen. And then going downwards at the midbrain, you also see the narrowing of the cerebral aqueduct, and also you get to appreciate the fourth ventricle, which, like I said, is situated between the brainstem anteriorly and posteriorly to the cerebellum. So don't forget the brainstem, you have the pons and the medulla oblongata. The upper part of the brainstem, which is the midbrain, also contains the narrow parts of that ventricles, which is the aqueduct of Sylvius. All right, so here the functions of the ventricles. The ventricles produce the cerebrospinal fluid and via the choroid plexus. So the choroid plexus are a network of blood vessels connected with some tissues of the brain inside the ventricles to allow for selectively allow certain fluid to only pass the junctions of the blood vessels. So that communication, or that joint, is referred to as the cerebrospine. That is referred to as the brain cerebrospinal fluid barrier, which is very important. And then the cerebrospinal fluid circulates, moves when they are produced mostly in the lateral ventricle. Most of it are produced in the lateral ventricle. They go into the third ventricle via the interventricular foramen, and from the third ventricle they move to the fourth ventricle via the cerebral aqueduct of Sylvius, and in the third and the fourth ventricle when going downwards into the central canal that goes all the way to the spinal cord, we have the three apertures. You remember we have the two lateral apertures, which are the foramen lucica, and then you have one in the posterior aspect and the midline, and it's called the foramen magendi. All right, so the function of the ventricles are mostly to protect, or to produce the cerebrospinal fluid, to allow for the circulation of the cerebrospinal fluid, as well as serve as hemostasis, because they help in balancing the chemical concentration of the brain by removing waste and bringing in more information. However, when any part of the cerebrospinal fluid is blocked, especially that interventricular foramen where the lateral ventricle connects to the midbrain, what happens is that there's going to be, because the choroid plexus continue to produce cerebrospinal fluid, the collection of this fluid increases, and gradually it tends to collect and increase the size of the brain and there the skull, because in the children they have this suture in the ventral part of their head. All of us have it, but in that on children, that is not fused together, and thereby allows for the cranium, or the calvary of the cranium, to actually expand with the expansion of the brain. However, we need to have a fast intervention to open, or to remove the occlusion, or remove the blockage, to allow for the complete circulation of cerebrospinal fluid. If not, if allowed, what happens is that the brain tissues tend to fuse with the skull or the calvarium, and thereby degenerating important neurons found on the cerebral cortex, and thereby you're going to have a child that is mentally retarded. So we do not want to have that. So early intervention matters. So next we are going to talk about this production of cerebrospinal fluid in the brain. And like we said, it is being produced by the choroid plexus. So in this image, you can see the numbers indicate where the movement as well. So you can see in number 1, you have the choroid plexus above the fornix. So in the midline, you can see you have the corpus callosum, then you have a fornix, that white matter. That is where number 1 is located. So above where you see number 1 is you see another pinkish structure, that is the choroid plexus, the blood vessels. So those blood vessels also allows for the production of cerebrospinal fluid to come into the lateral ventricle, and also in the third ventricle below the number 1, you can see another choroid plexus. These are also producing cerebrospinal fluid. So cerebrospinal fluid is going around, and then from there they move into the cerebral aqueduct of Sylvius, which is downwards, and then inside the fourth ventricle, that is located behind the pons and the medulla, and the pons superiorly, medulla inferiorly, and then posteriorly by the cerebellum, which is a small brain. You can also see another pinkish structure there just above where you have 2. So that also is a choroid plexus, also producing cerebrospinal fluid. However, the fluid are also circulating, and at that exact point where you saw 2, you see that that is where the cerebrospinal fluid flows out of the ventricles into the subarachnoid space. And we remember we have the subarachnoid space is the space between the subarachnoid matter of the meninges. So earlier on we saw the meninges, which is the dura mater, and then we have the arachnoid matter, and deep covering the brain's tissue is the pia mater. So between the arachnoid and the pia mater, we have the subarachnoid space. So now this blue, you can see these areas where it's indicated by blue. These are where the cerebrospinal fluid enters, and they enter via either the foramen lucica, which are the 2 lateral apertures, or the foramen magendi, which is the midline aperture. So after they go there, they gradually go around. So now the brain is swimming inside. Internally, it has cerebrospinal fluid, and externally, it also has cerebrospinal fluid. So this fluid is drained via arachnoid projection. So the arachnoid matter have projection at certain parts of the brain, and they are projected into the dura folds. So dura folds are referred to as sinuses, and they are the ones that drain the blood from the brain. So we don't have venous veins, but we have venous drainages, and they are via the folds, the double folds of the dura mater, which is the external meningeal layer. So externally we have dura, then we have arachnoid, then we have pie. So the dura mater, when it folds at different parts of the brain, it is referred to as the dural sinuses. And there you can see the projections of these arachnoid matter into these dural sinuses are referred to as the arachnoid villi or arachnoid granulation, depending on the textbook you're reading. So that is the point where these subarachnoid fluids are being drained into the sinuses, which then drains away into the internal jugular and then to the superior vena cava, to the right atrium of the heart. So here, the functions of the subarachnoid space, like we've said, they support, they protect, and maintain homeostasis, and also serve in transportation of nutrients, hormone, and also take away waste material away from the brain. So you can see how important they are actually. Then so here we're going to talk about the protection. So you can see this is the magnified view where you get to see the projection of the arachnoid. So the image on the image A is where we have the production of the cerebrospinal fluid, where you have the communication of the production with the coronoid plexus, where we have the formation, the production of the cerebrospinal fluid. And then B in that image is where you have the arachnoid granulation, or villi, which is where the cerebrospinal fluid drains away. At the bottom here you have what is referred to as the cribriform plate, where you have the nasal nerves. That is just an image just to indicate that. And then next you also have the communication between the subarachnoid space is just telling you how the blood vessels and also the cerebrospinal fluid is moving in the subarachnoid spaces. So like we said, production of cerebrospinal fluid is important, and on a daily basis about 500 mils of cerebrospinal fluid is being produced daily, and it gets reabsorbed back into the bloodstream via arachnoid granulation, so villi, and then they go back into mostly into the superior sagittal sinus, which is the sinus that is most found on the most superior parts and midline parts of the brain, and then gradually drains into the sigmoid sinus, and from the sigmoid sinus it drains into the internal jugular vein, and there it goes into the superior vena cava and then right atria. Okay, so that's the circulation there indicates the arachnoid granulation. So next, the components of the cerebrospinal fluid is water, electrolyte, glucose, protein, and also tends to have cells also moving inside that, mostly white blood cells. So the cerebrospinal fluid is important because we can also use them, we can sample them, we can take them, and we can do subarachnoid sampling to check for several conditions where you want to check neurological diseases, because you cannot take blood from the brain. So you can sample the cerebrospinal fluid to identify if there is any onset of any neurological disease or condition. So you use the subarachnoid space. Also, the subarachnoid space is also used as an anesthetic point for conditions for procedures such as a epidural injection. And next, we said the cerebrospinal fluid serves as protection because it serves as shock absorber, where whenever you shake your head, normally if the subarachnoid space is not there, your brain is going to be going right and left, hitting the calvarium or the skull. And so, like we said, it serves as it gives buoyancy, which makes the brain lighter in weight. So the pH value is also very important. So it also regulates ion balance, and also temperature regulations as well. So it also serves as a point of bringing nutrients and also taking waste away from the tissues of the brain. So normally, like we said, the clinical application, we use it to diagnose infection such as bacterial meningitis and viral meningitis, and including where we have the plasmodiums with the malaria. And then you can also detect neurological disorders such as multiple sclerosis, or you have a subarachnoid hemorrhage where there is a rupture of the blood vessels. And so when you take in the subarachnoid fluid from the cerebrospinal fluid from the subarachnoid matter space, you're going to see traces of blood. Therapeutic uses also the intrathecal drug administration. Like we said, we have the epidural, also the chemotherapy drug can be administered there. So now, what is the blood-brain barrier? So the blood-brain barrier is actually a tight junction, a selective point at which it's where the blood vessels are coming into the brain. So if you, on the most external part of the brain, you can actually see blood vessels on top of it. However, the blood are not going directly, but rather they serve as a point of transfusion. So you cannot get blood in the brain tissues. If you have blood in the brain tissues, then you're going to have hemorrhagic stroke. So how they exchange their nutrients or waste materials in and out of the blood vessels is that area is referred to as the gap, is referred to as the blood-brain barrier, and it is really reinforced. So it's made of several layers. So first, when you have the blood vessels, which is the most internal, in this upper image you can see it, which we have the basement membrane and the endothelial cells. So these are what give rise to the blood vessels. Then after, externally to the blood vessels, you have these green structures looking like structures. These are called the foot of the astrocytes. So the astrocytes tends to bind different types of cells in the brain together. They hold them together. So they also help, the foot of it helps to reinforce the gap junctions, making it very narrow to allow only selective nutrients and ions to move across the brain and the blood vessels. Other structures also reinforcing this is you have the pericytes, and the pericytes also are important, as well as the microglia cells. So the microglia cells helps to hold all the structures also in place, and those are mostly the cells that makes up the gap junctions. So the gap junctions, this barrier are important because they serves as a protection. They challenge, but it serves as is challenging because it doesn't allow for drugs to pass through it, and that is why a lot of neurological degenerative diseases are difficult to treat. And also, disruption of this blood-brain barrier could lead to several neurological diseases because you cannot control, as well as brain tumors as well. So the formation of blood cerebrospinal fluid barrier, you remember when we talked about the choroid plexus. So the junction between the choroid plexus and the ventricles is what we refer to as the blood cerebrospinal fluid barriers. And this is because you also need to selectively allow certain things from the blood vessels or the capillaries come into the ventricles, and also the endothelial cells of the choroid plexus, which are the blood vessels, are also lined externally by tiny cells, which are called the ependymal cells of the ventricles. So these ependymal cells of the ventricles assist in making the junctions or the spaces between the endothelial cells very tight, allowing for just selective structures to move in onto the ventricles. So other factors that assist also are the basement membrane of the choroid plexus. You remember the blood vessels also have externally, they have the blood membranes. Then we also have transport proteins, which are like gap junctions that allow for selective crossing of structures. So the clinical significance of blood cerebrospinal fluid barrier is also to protect our protection of the brain, that is restricting the entry of any harmful substance in and outside of the brain, maintain a stable environment. It's also challenging, just like the blood-brain barrier, for treatment of drugs, and also disruption of this could also lead to different types of neurological disorders. So now we're going to be going into the next study, which is the spinal cord and blood supply to the central nervous system. So this is study 4. So the spinal cord is located inside, just like we talked about the brain, which is having several protective layers. This spinal cord is also having protective layer, and it is protected by the vertebral bones that extends from the base of the foramen magnum and all the way to the coccyx. So we have several bones that form that. So we have 7 cervical bone, 12 thoracic bones, we have 5 of lumbar, and then we have 5 fused sacral bones. So the 5 fused sacral bone are the bone that makes the posterior part of our pelvic bone, and then at the bottom we have our tailbone, which in us has shrinked and gave rise to what is called the coccygeal bone. Mostly 3 small fused bones, but in other animals like dogs, you can see them because they have a tail. You can also follow to see the vertebra. We also to eat cow tail, we can see it also has a vertebra, but in us, because our tail has been degenerated during our development, and that is why we do not have the tailbone, but rather a little bit of just showing that there was once and that tail, but has gradually rudimentary. So these tends the vertebra also provides support protection for the spinal cord. So the spinal cord is almost hanging inside the vertebral bone. However, we have the dura and the meningeal layers here as well. So you also have the dura mater, you have the arachnoid, and also the pia mater is lining directly over the spinal cord. And it's very important because initially we have the spinal cord extending all the way to the coccyx, but because of the elongation of our body grew, and that is why we also have the growth of some nerves. And in image on the right hand side, you can see the lateral view of the spinal cord and the posterior view of the spinal cord, with nerves root leaving the spinal cord. So these nerve roots indicate so you can see we said that we have 7 cervical bones, but we have 8 cervical nerves. This is because initially the cervical nerves were coming off superior to the vertebral bone, but after the 7th cervical bone, where we had another nerve coming below it, which is the 8th, then we started having the thoracic bone has thoracic nerve 1 coming below it and all the way downwards. So you can see that the green area, the green nerves at the lower part is lumbosacral, and it is important because the spinal cord tends to finish at L1. This is very, very significant because you do not want to have the spinal cord extending beyond that, and this gave us the advantage so that we can actually put an injection, or where we do the lumbar puncture, we can inject anesthetics and also take up some the cerebrospinal fluid at this point, because we are not going to injure any spinal cord, because spinal cord, when injured, it's hard for them to heal, and when they heal, they have scar, which could also lead to paralysis at different parts of at the area where the new cell bodies of those neurons are coming from. So as we are growing, our body also went into curvatures, and these curvatures are important to maintain our status. And because we have several areas, our skills, our fine skills are found mostly on our upper limb, that is our hand, where we tend to write, we knit, we paint, we dance, we do a lot of things with fine skills with our hands. And that's why the nerves coming from the cervical area and upper thoracic are going to combine together to give rise to what is referred to as the cervical plexus. So this cervical plexus are where you're going to have the different nerves coming to innervate different parts, the muscles, and it's also cutaneous innervation for our upper limb. And like we said, our upper limb is a very skilled area where we need lots of more neuron or control for our fine skill, and that is why at that spinal area you have what is called the spinal expansion at that point. And then also because of our lower limb, we tend to have another lumbar enlargement. This lumbar enlargement is to assist in the formation of the lumbosacral plexus, which helps in the innervation of our lower abdomen and lower limb. So this is the curvature that we gained as we are growing up. So the first one, with the first one we have in the cervical curvature is important. You remember when the baby is born, their heads keep shaking, but the moment they have the cervical curvature is formed, the head stops shaking. And then you also have the thoracic curvature, which helps to move back and give room for the location of our thorax, for our lungs and our heart as well. Then you have the inwards curvature of the lumbar area, and this is important because that curvature allows for the distribution of our weight and allows the baby to sit, and also allows for the equal distribution of the weight when we stand up and to walk. So next, why the importance of this curvature is it supports it provides a structural support to our body to reduce stress and also distribute the weight, allows us to be very flexible, and also it serves as a shock absorber. And then we have abnormal curvature. We tend to have abnormality in this curvature where we have a hyper thorax curvature, which is called the lordosis, and also you have the hypercurvature of the lumbar area is called the lordosis, while when you have an extended thoracic curvature, it's called the sclerosis. And then when you are abnormally curved away from the midline, we call it the scoliosis. So like we said earlier on, we have 31 pairs of spinal nerves, and that is C1 to C8, T1 to T12 thoracic, and then we have L1 to L5, F5 sacral, and also we have one coccygeal nerve, which is also very important. In the spinal cord, the gray matter is more inside while the white matter is on the exterior. This is the other way around when we talked about the brain. So here you can see that the spinal cord ends as a cone structure at that point where at the bottom there where you see the lumbar, where around the yellow area, just the junction of the yellow area and the orange area, you can see where the spinal cord ends at the level of L1, L2, and that is called the conus medullaris because it looks like a cone, inverted cone. And then the other nerves comes down, so you have L1, L2, L3, L4, L5, S1, S2, S3, S4, S5, and coccygeal 1 all come down as a bundle, or like that looks like the tail of a horse. And because they look like the tail of a horse, that is why they are referred to as the quadra equina, which is basically the tail of a horse. And then the actual part of the spinal cord also ends as a narrow terminal structures that goes to fuse onto the coccyx, and this is referred to as the frenulum terminale. So now you get to see that the midbrain also has some certain features. It has a cervical expansion or enlargement. It has a lumbar enlargement. The spinal cord terminates at level of L1 and L2 to allow us to be able to inject or take do a lumbar puncture to either inject for anesthetic injection or to acquire cerebrospinal fluid for testing, take a biopsy for testing as well. So everything below L2 is safe for lumbar puncture, but it will be good to also have to scan to be sure that there is no any abnormal extension of the spinal cord in that particular individual that may serve as a variation. So here in the image, the back part of the vertebral bones has been removed, and then you can see the quadra equina coming down as it, the name serves as the tail of a horse. So here is a particular point where you can actually do lumbar puncture. Because you will not endanger or injure the spinal cord. So here, cross section of the spinal cord is this, as we have the view of it, and then you can see that most externally you have the vertebral bone protecting it. After the vertebral bone, you have the pink, the external hard pink is the dura mater. The inner pink, you have the arachnoid mater. Then between the arachnoid mater and the pie mater, which is the one covering directly on the spinal cord, it's called the subarachnoid space as well. It also contains the cerebrospinal fluid. Then you also have the spinal cord, where in the middle you can see a H-looking area structure. The H-looking area structure is referred to as the gray matter. Like we said, in the brain, the gray matter is exterior, while the white matter is internal. But in the spinal cord, it is the opposite, where the white matter is more on the exterior, while the gray matter H is in the interior. So these are the expansions, and also you can see how it extends, and from the foramen magnum, and extends all the way to become the conus medullaris, and they form the tail of a horse, which is the quadra equina, while you have one filum terminale which goes to fuse on the coccyx. So here it is the superior view of the spinal cord, the one on top, and the image on the bottom is a lateral view of the spinal cord, and so you can see it with also the subarachnoid space containing the cerebrospinal fluid. So here is also an image that illustrates that particular aspect, where you see the dura mater on the exterior arachnoid mater, because it looks like a web, cobweb thing. That is why the name arachnoid mater implies. And then you can see the pie mater is directly on top, lining the spinal cord proper. And the spinal cord is also also has blood vessels. So it has one anterior spinal artery and two posterior spinal arteries, and then it also have two spinal veins in the anterior aspect, as well as two spinal veins in the posterior aspect. But one thing is that I want you to know that at every level, we said that we have spinal nerves, that is nerve fibers leaving the brain. So you remember when we talked about the 31 pairs of spinal nerves, they are coming up from the brain, from the spinal cord at different parts. But note that spinal cord is also part of the central nervous system. So the clinical significance of the spinal cord is very important because any injury, like we said earlier on, could cause scarring, and scarring, because the central nervous system does not regenerate and does not reculture itself, just like when we injure ourselves, where we have it can actually come and sometimes you don't see a scar, but in the central nervous system it does not do that. However, in the nerves, that the peripheral nerves, we can have regrowths of the regeneration of the nerves, but in the spinal cord for now we cannot actually do that. However, scientists are trying to discover if they can use stem cells to rejuvenate or regrow some parts of the brain, injured brain. So for now, if you have an injury at any level, you're going to have a paralysis of the structures that are being supplied by nerves coming out from that level. So we also tend to lose the sensory information that comes in from that particular level, and also there's going to be disruption of autonomic functions as well, and also we're going to have the nerve spinal shock, where temporarily we're going to have temporal paralysis. So these are some of the significance of the spinal cord, where one, we can actually use it for advantage to get amniotic fluid, not amniotic fluid, but rather cerebrospinal fluid from the brain to test it for any kind of condition, hemorrhage, or any early neurodegenerative diseases. So we can use the lumbar puncture. We can also use that lumbar puncture for anesthetic administration, like the epidural. And the next image, where you're going to see an injury of the spinal cord, or we have a the herniated disc, where the intervertebral disc, that is the disc, the cartilage, that cartilage chewy looking bone found between the vertebral bone, that is called the intervertebral disc, tends to move backwards and suppress on the nerves exiting the intervertebral spaces. So between one vertebra and another vertebra, there's going to be a space where that spinal nerve lives. So sometimes you're going to have a condition. So most of the time, when you stress your back a lot, gradually what is happening is you're going to have a disc slide, where the intervertebral disc tends to be sliding backwards a bit, and after it continues to slide, it comes to impinge on your spinal nerve, and that by injuring your spinal nerves, you're going to be having so much pain, and even in moving and walking. But now people can actually relieve this pain by a kind of surgery. Another is when you have the spinal stenosis, where you have an injury or a trauma which could lead to compression of the spinal cord, and sometimes damages or injury to the spinal nerves, where you compress your spinal nerve, and if it's excess, it leads to paralysis as well. Another condition is where you have so now we're going to talk about the arterial supply of this brain and spinal cord. Just initially we kept talking about the spinal anterior spinal artery. So before we talk about the conditions associated with the blood vessels, like the hemorrhage, let's talk about the blood supply as well as the venous drainage. Then we can understand when we talk about the clinical application for that. So the brain, like the first image on the left hand side, is the base of the brain, where you can see two arteries from the bottom, because the two arteries fusing together to give rise to a big artery. So those two arteries coming up to fuse together are called vertebral arteries, and by the time they fuse together, they form what is referred to as the basilar artery. So the second image is just an illustration in depicting that. So this basilar artery gives off some branches. So the moment it fuses together, it gives rise to the inferior posterior cerebellar artery, that is the inferior posterior cerebellar artery. And then just as it starts to ascend, it gives rise to the anterior inferior cerebellar artery, and then as the basilar artery continues to ascend, it gives rise to smaller arteries, which are called the pontine arteries. And then at the base, just at the end of the pons and the superior aspect, it divides into two. So when it divides into two, it gives rise to the posterior cerebral. This is the big brain, posterior cerebral arteries. So when it gives rise to the posterior cerebral arteries, this posterior cerebral artery is going to innervate or supply the parts of the temporal bone on the lower aspect, as well as the occipital lobe. Then another artery came in. You remember when we talk about the internal carotid artery? So the internal carotid artery comes in from the foramen as well and enters the cranium, and then what happened is it folds over itself, giving rise to one branch that goes to the ophthalmic artery and another branch that goes anteriorly. So the moment internal carotid artery comes in and folds, giving rise to those branches, it continues at the middle cerebral artery. Middle cerebral artery, not cerebellar, but cerebral artery. So the middle cerebral artery goes to innervate more of the temporal lobe, as well as it communicates with us to the inferior aspect of the front, the parietal lobe and the frontal lobe. And then, like we said, one artery is going off, giving rise to the ophthalmic. Another, we said, give rise to two branches. So the other artery goes anteriorly, and then they also they become very close. They lie close to each other, and they give rise to what is referred to the anterior cerebral artery. This anterior cerebral artery is going to go and supply the lower part of the frontal lobe, the medial part of the frontal lobe, and superior part of the frontal lobe. All these three arteries, the posterior cerebral coming from the bifurcation of the basilar artery, the middle cerebral artery, which is after the internal carotid artery, comes to fold over each other and give rise to branches. The main branch continues as the middle cerebral artery, which innervates more of the temporal lobe and also superiorly the parietal lobe. And then we have the other branch of the internal carotid goes anteriorly, all right, anterior to the optic chiasma. So when it goes anterior to the optic chiasma, line side by side, the one on the right and the one on the left, lies side by side and goes in to innervate the frontal lobe, the inferior part of the frontal lobe, and also superior part of the frontal lobe is called the anterior cerebral artery. So, and then there's something important that the anterior fibers, okay, the anterior cerebral artery tends to send in a branch that communicates with each other. This is referred to as anterior communicating artery. It's referred to as the anterior communicating artery. While the middle cerebral and the posterior cerebral, you remember the posterior that came from the bifurcation of the basilar, and the middle coming from the internal carotid, they also tend to communicate with each other via what is referred to as the posterior communicating artery. So these branches, all these anterior cerebral communicating with the anterior communicating, comes backwards, it comes out from the middle. So then you have the middle communicating with the posterior via the posterior communicating artery. These communications between the posterior, middle, and anterior give rise to what is referred to as the cycle of Willis. So they are very important because they give rise to the innervation of the deep structure of the cerebral cortex and also the cerebral cortex itself, while the vertebral artery that comes to fuse together give rise to the posterior, inferior, and superior cerebellar arteries, while just before the basilar artery bifurcates, we also have the anterior cerebellar artery. All right, so another artery that we're going to talk about is, if you notice in the first image, before the two vertebral artery fuses together, they give rise to a narrow artery indicated by green here. So in this image you can see it, they are also in the midline as well. That is the anterior cerebral artery. Anterior spinal artery. An anterior spinal artery is the one that runs on the anterior surface of the spinal cord, which we can see it on the third image. So the third image is showing us the anterior cerebral artery, and then it's also having tributaries or contributions from different arteries. Some of these are the ascending cervical arteries. We also have the anterior segmental arteries. We also have the pi plexus, and also have the great radical arteries. They are all contributing to the anterior spinal artery. So these are also another set of image which indicates or shows the cycle of Willis. So you can see the cycle of Willis is the bifurcation of the basilar artery, which is the posterior cerebral artery, and then we can see the middle cerebral artery coming from internal carotid artery, which give rise to ophthalmic artery, and another artery which is the anterior cerebral artery. So, and then between the posterior cerebral and the middle cerebral, we have a branch which is the posterior communicating, indicated by those pink area. At the first image we referred to cycle of Willis, and then you can see that you have the middle cerebral is the one that gave rise to the anterior cerebral. So you can see the two anterior cerebral artery are lying very close to each other, but they have a very short communicating branch, which is the anterior communicating artery for that. So that forms the cycle of Willis. All right. So if you go to the spinal cord, like we said earlier, the spinal cord has one artery in the anterior aspect and two arteries on the posterior aspect of it, and you can see that both of them are communicating, where majority of the anterior horn is the anterior horn and also part of the posterior horn of the gray matter are supplied by the anterior cerebral artery, while most of the posterior fascicles or the white matter are innervated by the two posterior cerebral arteries. So now we're going to talk about the venous drainage. So the venous drainage are mostly by what is referred to as dura sinuses. So the dura sinuses are found within the fold of the dura mater, and the dura mater is a covering, like we say, is the most external meningeal structure found on the most external aspect. And when they fold when you have this mid sagittal flexure, when it goes deep inside, it forms two folds. Okay, in between these two folds, you're going to find the superior sagittal sinus and also inferior sagittal sinus. So here in the second image, the brain tissue has been removed so that you can appreciate the dura sinuses. So the first superior sinus, which is the most superior, and you can see it in the first image as well. On the left hand side, you can see it running on top of the two cerebral hemispheres, and it actually comes to connect with inferior sagittal sinus. So the inferior sagittal sinus is deeper, we cannot see it much, so it connects via what is referred to as a straight sinus. So we're only concerned with the major ones. So the straight sinus, if you look at the image 3 and top, you see the openings of the straight sinus. So the straight sinus is communicating not just with the superior and inferior, but rather you also have one and coming from the inferior aspect. If you look at the third diagram, you see on the right hand side, you see the occipital sinus located in between the two indicated inside the flex cerebelli. And then you're also going to see the occipital sinus comes to drain into the trochlear trochlear notch. So this is where the superior sagittal meets with the straight, and then you also tends to have the transverse sinuses. So the transverse sinus, like the name implies, they are found on the lateral planes, and then they come in to drain into the sigmoid sinus, and from the sigmoid sinus they drain into the internal jugular vein, and from the internal jugular vein they drain into the the right atrium via the superior vena cava. So next, we're going to talk about the autonomic nervous system. The autonomic nervous system are important because they have several functions that are importantly divided into 3. We have the sympathetic, parasympathetic, and then we have the enteric. So enteric is the one that allows for the relaxation and contraction, because we cannot control it. Relaxation and contraction of muscles, smooth muscles, especially those found in the digestive system and respiratory system, that allows us to contract the tubes and relax them in digestion, and also bringing in oxygen and releasing of carbon dioxide. Sorry. So here the functions is the control in the heart rate and blood pressure, respiration, digestion, body temperature, because they control the hair follicles. We have muscles at the base of the hair follicles that when you are cold, they tend to rise. When they rise, they increase the action potentials in our body, and then we try to emit heat from our body. And metabolism, that is our digestive system, also assists in that. The dilatation of our pupils, so in our eyes, where we have that dark coloration of the structure in the parts of our eyes, when you look deep into your eyes, you see something like it's hollow in between your eyes. That hollow area is known as the pupil. And when you are scared or frightened, what happens is that the diameter of that pupil increases. And okay, so that is dilatation of the pupil. Is also being controlled by the autonomic nervous system. All right, also sweating, similar to what is happening with the hairs, the sweating also occurs and allows us to retain the temperature, reduce the temperature of our body. Also sexual arousal as well, and other functions. So we have, like we said, the major parts of the autonomic nervous system is sympathetic and parasympathetic, but we also have the enteric, like we said, casually controlling the contractions of smooth muscles in other parts of our body. So the sympathetic nervous system is a system where it controls your flight or fight. It's either if I scare you, you stop and fight me, or you run away. That is what it means. So sometimes if I scare you, what happens? All your pulses increase, your hair rises, your eyeball dilate, you are very angry, all your hormones are just being angry. Okay, and then what happens is that your heart rate tends to increase. You're going to you have a contraction of several parts of your body because you want to either beat somebody or you want to run. You're also dilating the pupil. You're also dilating your air sacs, that is your respiratory system. You're taking in more oxygen. That is why you are breathing heavily. Okay, all right. So you're also releasing so many glucose. That is why sometimes if you finish fighting or you're angry or you get very hungry, also it promotes the release of adrenaline and noradrenaline, and this causes you to sweat, the contraction of blood vessels, okay, in the skin and guts. All those are all contracting. However, the opposite of sympathetic is the parasympathetic, where after you have finished fighting, you go and sit down. What happened? That is rest and digest, where you're going to digest the information. What just happened? Why did this happen? Do you understand? That is where you now start sometimes sweating. You're going to have slow heart rate. You're going to call, your people are going to become smaller. If sometimes you get angry and start crying, your airways is now constricting. That is why sometimes if you finish, you relax, then you start saying, I cannot breathe. Some people would faint. Now you feel like, I want to go to the toilet, I want to pee, after you have finished fighting or running away. Okay, you also have the promotion of the saliva and digestive enzymes. You feel very hungry, like your stimulation of urine and defecation, okay, and facilitating sexual arousal as well. So now we talk about the enteric nervous system. Like we said, it's important because it serves as motility to digest our digestive system, the secretion of digestive enzymes and hormones as well. It increases the blood flow to the digestive system, also communicates with the brain about the gut environment. So anything we eat, is going to be affecting our brain. So that is why our gut is our behavior. So some people now, researchers say, what you eat is who you are. So this is basically how it relates. So the so these are some of the structures or systems or organs that are being controlled by the sympathetic nervous system and also the parasympathetic nervous system. And then the red line is telling you which are, because we have fibers coming from different parts of the body, the nerve fibers, and they also goes to do this innervation. So the pink color is telling you that sympathetic controls your eye, which is the dilatation, lacrimal gland. Your eyes are wide open, and then you can also see salivary glands. And the thing is, any any effects, any sympathetic effect you have on organ, you definitely have a parasympathetic effect from it. So that is like what we just said initially, where all those you're going to see your hair follicles have risen, oil and everything is being produced. So these are some of a summary of what the parasympathetic does, control, and the and sympathetic controls as well. So now we talk about the clinical applications of the central nervous system regarding the blood vessels. So if we have a rupture of the blood vessels, and now a lot of Nigerians are having that issue, so we need to understand what is causing this rupture. One of the most common cause of this rupture is aneurysm, where a part of the blood vessel swells and then gradually bursts, allowing for blood to seep into the brain, to come in contact with the brain tissue, which is called the hemorrhagic stroke. And then you also have the blockage of the blood vessels, and this causes an ischemic stroke, and you can see those are in different illustration as shown. However, now when you have arteriosclerosis, that is fat deposition in your blood vessels or plaque, all right, sometimes you have what is called the embolism, where you have air in your blood vessels and goes and to the heart, is going to cause a kind of a shock as well to the person. So that is why try as much as possible to remove the airs in the needle. And sometimes air goes into the blood vessels via, maybe if you have ever had a fracture and then there was a healing, or sometimes you have an opening and the air entered into the blood and the bone marrows, and sometimes gradually this air tends to gradually move and enter the blood system. So this is what is called the embolism. And so it is very important to identify this and differentiate it to give rise to do the appropriate treatment. So here you can also have sinuses with thrombosis, and then you can actually treat this thrombosis by using what is called a mesh wire, and you pull the mesh wire in it, because the mesh wire, the thrombosis can actually block the blood vessels. So you put a mesh wire, pull the mesh wire, it tends to pull away out the thrombosis you have in any of the blood vessels, including the sinuses. So thank you very much. I'm sorry for the long lectures. It's a long slide I needed. You can actually keep watching it half by half, but if you have any question or comments, please, you can actually ask it in the WhatsApp group, or you can drop a comment here. Have a good day.